All right, so just as we looked at the changes in potential that occur across the membrane last time with the autorhythmic cells, now we're going to look at the contractile cells. So this is very similar. And remember, these changes or fluxes in current that we are looking at here, they're what is changing in the inside of the cell relative to the outside. Sometimes we're becoming more positive compared to the outside, sometimes more negative. So whereas last time we were getting these changes in autorhythmic cells due to funny channels that were opening upon hyperpolarization, now we're going to be looking at the changes that happen in contractile cells when that depolarization from the autorhythmic cells reaches those contractile cells via gap junctions. Okay, so the first thing we're going to have then are just our axes like we had last time. All right, so we got a y-axis and an x-axis. Of course, this is going to be membrane potential. We measure that in millivolts. And then down here we have time, and we measure that in milliseconds. Okay. Just some things we can plot out here. This will be plus 30 at the top of our axis here. Let's say our threshold, that is going to be at negative 70 for these contractile cells. And we're going to go as low as negative 90, right, when we're polarized. Okay, so now let's add some of our proteins or channels to this figure. And I'll try to color code everything here. So let's do it this way. Put this off to the side up here. Let's go ahead and say that blue, everything we put on this figure in blue, that's going to be a calcium-related event. So Ca2 plus. Let's say that green, is going to be potassium related, and let's say that red then is going to be sodium. Cool. Okay, so the first thing we're going to start out with then is a little bit of green. And we're actually not going to start at the first step here. It makes a little more sense just to start at the fourth so we see what's going on. So here we are. We're going down at, when we are polarized, down at negative 90. Okay. And this is going to be our resting membrane potential here, around negative 90. Okay. It's going to be a little more negative than we're used to seeing in things like neurons, for example, because these potassium channels here are leaky. So I'm going to call this K and a little sub L. So we'll put a little 4 above this. And because we have potassium leaking out, right, remember that's a positively charged ion, so when that's leaving we become more negative. So we're down here at negative 90, a little more negative than usual. And let's go up here and start like that. So this is step 4. And 4, we're going to have our potassium channels, K plus, leaky. These are going to be open. And that's where we get this resting membrane potential of around negative 90 millivolts. But then we have that incoming depolarization from those autorhythmic cells. Okay. Now, this is going to involve a sodium flux. So we're going to have a voltage-gated sodium channel that is going to open now. So we're going to see this rapid depolarization. We're going to go all the way up to positive 30. Okay. And if that was step 4, now we're going to start, and we're going to call this just arbitrarily step 0. So we're going over here, and let's write out what's going on at step 0. Well, this is now voltage-gated sodium channel that is opening. We get that incoming depolarization from autorhythmic cells that triggers the opening of these voltage-gated sodium channels. Remember, they're voltage gated, so that depolarization is going to cause them to open up. Okay? Now we're back to our voltage gated sodium channels acting in more of a classical fashion, right? That's opening upon depolarization from that incoming signal. Not like with our funny channels when they're hyperpolarized, they open. So what we see with this is a rapid depolarization all the way up to positive 30 millivolts. Okay, and why does that happen? Well, the sodium rushes into that contractile cell. So we have a lot of positive charge moving into that cell now relative to the outside. Okay, now we're going to involve another potassium channel. And we're going to see this small repolarization here. And we're going to call this step 1 now accordingly. And this is going to be K plus T. Oh. So that's a small TO there if you can't read it. And that means transient outward. Okay. And I am going to go back here real quickly and just label this. Since we put it everywhere else, this will be Na plus. Back to potassium here. So at step 1, we have this transient outward potassium channel. So that is a voltage gated K plus transient outward. That is going to open. Now again, when we say transient, that means kind of short-lived. So it's only open for a short period of time. And you can remember outward, just that's the flux direction that the potassium is going to be leaving the cell. When that leaves the cell, we get a little bit more negative, right? So we start repolarizing a little bit, and that's where we see this decline. Okay. Now the other event that you can note that happens here simultaneously, and I'll try to color code this correctly, will be those voltage gated sodium channels from before are going to close. So voltage gated Na plus close. And because those close, then we stop having positively charged sodium ions entering the cell, and that's how we start to decline here and return back to polarization. All right, next we have a calcium channel. First time we've looked at those here today. And this is going to be event 2. And here we're going to have just this plateau. So this is event 2 now. Involves calcium. You see A2 plus. So here we have these voltage gated L-type calcium channels open. Remember that L stands for long. They're open a long time. And we also, at this step, have our voltage-gated potassium transient start to close. So if we look at these two events here, I kind of explain why we see this plateau. Remember, a plateau is just a flattish area here, and that's what we see here. So if we think about this, it makes sense, right? We have our potassium channels that are open. So potassium is, because it's been concentrated within that cell, via the sodium potassium pump, it is leaving when we add a little channel for it there in the membrane. So that positive charge is leaving, we start hyperpolarizing. Not hyperpolarizing, repolarizing. Okay? But then we have some calcium channels open. And calcium then is going to move into the cell. So that's a positively charged ion moving into the cell. So you have these two things that are fighting each other here, right? you have potassium leaving, so becoming more negative, but calcium entering, so the cell's becoming more positive. And they kind of cancel each other out, and we get this plateau, where it's just kind of flat for a while. Okay? So that's how we have that plateau. And then we have our third event here, which, again, is going to be related to potassium. Let's see this. Depolarization. So this, I'm sorry, repolarization. And this is going to be step 3. At step 3, we have these voltage-gated L-type calcium channels. Well, they're now closed. And because they close, the positive charge with those calcium ions stops entering the cell, right? And if that stops, then we become more negative, especially when we have these now voltage-gated potassium channels. I just call these ordinary, the ones we're familiar with. They open. Okay, so this is another type of voltage-gated potassium channel. They open, which means a bunch of potassium is now rapidly leaving that cell. So I'm going to put K plus out. And if that's leaving that contractile cell, now we become negative again. We repolarize. And we're allowed to do that because those voltage-gated calcium channels, where calcium was entering the cell, are now closed. Okay. Last thing I'm going to add on here, I forgot to add it, is our threshold at -70. And that's just when that depolarization from the autorhythmic cell allows us to hit -70. That's when we get this action potential. Okay. And with these two events, this allows for a repolarization. And we approach -90 again. The very last thing I want to point out here, let's do a new color, how about some orange, is that if we were to plot a contraction of a contractile cell on this same figure, we would have different axes, but it would look something like this. So that plateau corresponds kind of to the length of a contraction. Why is that? Because in between here, that gives us an absolute refractory period. And we'll talk about the significance of that next. Basically, we cannot get a contraction during that plateau phase no matter what we do. And that is going to be very important for the contraction of the heart. Let's take a look at that.